Thank you, everybody, for attending this panel discussion about the documentary Human Nature, hosted by Cold Spring Harbor Laboratory. I'm Elliot Kirschner, an executive producer and producer on the film. This film was produced by the Wonder Collaborative, a new initiative to innovate science films for the general public. My own background is in news and documentaries, including a longtime collaboration with Dan Rather, a news icon, who also served as an executive producer on Human Nature. In fact, this project began many years ago with an interview between Dan Rather and Jennifer Doudna, who's joining us here today, and likely needs no introduction. But nevertheless, Jennifer, if you would please introduce yourself.
Thank you, Elliot. It's a pleasure to be here. My name is Jennifer Doudna, and I'm a professor in the San Francisco Bay Area. I run labs at UC-Berkeley and the Gladstone Institutes.
Great. And also joining us, Alta, Charo, do you...
Hi, Alta Charo. I'm professor of law and bioethics at the University of Wisconsin.
And Zach Lippman.
Thanks, Elliot. I'm a professor of plant biology at Cold Spring Harbor Laboratory.
Wonderful. Well, we're going to jump right in. I mean, one of the things that this film is about is the fact there's a huge story in science, a story that when we started this wasn't getting us enough attention. Still gets a lot more attention now. But since that time, there's also been a whole other, obviously, story, a tragic story that really deals with science around COVID-19. I think one of the things I get a lot of questions and sort of like to address to you, Jennifer, is sort of, you know, where does CRISPR and COVID-19 come together? In what ways? You know, there's a technology that's about viruses, and this is a virus. And we hear about testing. Sort of, maybe if you could give us a sense of where that is now. And
sure. Well, first let's just define the technology we're talking about. So CRISPR is an acronym that represents a bacterial immune system. It's a way that bacteria can avoid viral infection. And in fact, the biology is very interesting. It actually is a pathway for microbes to find and destroy viruses that are in the process of infecting them or could infect them and then provide protection in the future from those viruses. So it's actually a system that is, a natural antiviral. And in the time of a pandemic, I think this, resonates with all of us. Is, wouldn't it be great to be able to use a system like this to help us fight the virus, or certainly at least to detect it? And I think that, and I know we're going to get into the other things that the technology is being used for, but I think one of the extraordinarily interesting things about what's happening in science right now is that there are many research labs, both academic and companies, who are pivoting to use CRISPR as a way of detecting the coronavirus, and in fact developing tests that could provide very rapid diagnostics that allow sensing of the viral genetic material in a way that is very reminiscent of how this works in bacteria.
I think one of the things that makes this CRISPR such a powerful gene editing tool is sort of the specificity that nature, through billions of years of evolution, has derived. So how does that sort of basic science, how is that harnessed towards sort of this testing that you talked about? And might it also become a tool in terms of treatments as well as diagnostics?
Yeah, well, for diagnostics, it's actually used in a way that's very similar to how it operates in bacteria, namely to find a particular piece of genetic material from a virus. And for those that are a little farther down in the weeds, coronavirus is an RNA virus. That's its genetic material. And there are some types of CRISPR systems that are definitely, programmed naturally to identify RNA sequences. And so we use it in the same mode as a diagnostic to find coronavirus RNA molecules. And this can be done in, not just in cells, but it can be done in saliva or, nasal swabs, the kind of samples that are being taken for testing with other kinds of testing formats. And then in terms of using it as a therapeutic, it's an appealing idea that I think in practice will be difficult because it would require delivery of these CRISPR molecules into patient cells. And as you can imagine, when a patient is very ill with coronavirus and has, for example, infection in their lungs, and where it might be valuable to get an antiviral system to that point in the body, unfortunately, it's just very difficult to do that kind of delivery right now and get it into every cell where it would be needed. So I personally think that, at least in the near term, this system is going to be great for diagnostics. And then, when we think down the road of how does science get ready for future pandemics, there's where I think, there could be additional technical advances that allow CRISPR to be deployed in a therapeutic fashion in the future.
Wonderful. And I want to add, as we go through this, we do have a Q&A function in the bottom, and please populate that with questions. I mean, there's no shortage of things to talk about. We really want to, CRISPR is such a diverse and interesting topic that we really want to make sure we're answering what people out on the audience are interested in. But one of the things that we did in this film, in Human Nature, and you can tell from the very title, is that there's a lot of questions about what this means for human evolution, and we try to frame this in the sense that CRISPR could change our ability to edit our own genes and what that means. There's a real question about what the time horizon is for that. Where everything is much faster is really in other uses, so I would like to sort of ask Zach to jump in on this, and Alta. These are things that we've talked a lot about. CRISPR is being used in agriculture already. It's being used in other forms besides sort of the human, Brave New World, which we'll also talk about here too. But I just think that there's a sense here about how readily available this technology is. In some ways, where it's already being used is maybe gets less attention than it really should in the popular press. So maybe, Zach, you could talk a little bit about where you think where CRISPR is being used already in your research.
Sure. So we, as well as many others in plant biology, are using CRISPR extensively. And the initial desire to use it was as a tool to study gene function. So the ability to identify any gene that you'd want to study its function, you'd want to what's called knock it out. And that essentially create a mutation, a targeted mutation. But it quickly became apparent that there were application values to using CRISPR. And my lab, as well as others, including Cold Spring Harbor, Dave Jackson, who works on corn, have been using CRISPR to modify traits that are tied to agricultural productivity. And I would agree with you. I think the pace of innovation in plant biology for using CRISPR, both in fundamental biology as well as applied science, is really moving very quickly. And I often describe it as a freight train that really can't be stopped. And some people don't like hearing that because those who are anti-GMO are always worried about the relationship of what CRISPR is and its technological use in agriculture compared to the genetic engineering that was pioneered in the mid-1990s. But it's here to stay, and it's extremely powerful, and we're using it to, for example, one case where I think we're making a lot of headwind is showing how we can convert very tall, unwieldy growing tomato plants into plants that are highly compact and very rapid cycling, and they can be grown in urban agriculture settings. So that's just one example among many where it's being deployed in agriculture.
And Alta, you and I have talked about this many times, and one of the things you've told us about the film is, while you're a big fan, you feel there should be many other films also about CRISPR and things that tackle many different applications on it. And I think there is this question about GMO, about regulation, about how this technology, how fast it's coming. How do you explain to people when they ask questions? You've followed this for long before CRISPR and sort of seen this sort of field develop really rapidly in many different forms of our ability to edit genes. How do you sort of address the timeline question and the application question? How fast is this going to happen? Where is it happening? And should one be scared about that?
I don't think the technology alone can explain what the timeline is going to be. It's also about whether there's some crying unmet need out there that's driving people to use it. So, for example, when Zach talks about its use in agriculture, we can imagine lots of places where there might be interest in having crops edited to deal with the changing climate or to reduce the number of acres that have to be devoted to agricultural practices that might deplete the soil, or to make foods more nutritious, or to make them growable closer to an urban center with a smaller carbon footprint. So first you have to have a pull mechanism. You have to have people that are out there waiting for something, right? And the second thing you need to have is some kind of industry that's going to be able to funnel money at an industrial level to take things from laboratory through that kind of intermediate pilot stage and put it into widespread practice. And last, it's going to be a regulatory thing. One of the differences between the human applications and the kind of ag applications Zach mentioned has to do with the regulatory regime, which for many, not all, but for many engineered plants is a lot easier than the regime that's applied to anything that's a human therapeutic that will be directly transplanted into the body in the form of edited cells. And so that makes it possible to imagine a business plan that will result in some return on investment within one's imagined lifetime. But when it's talking about editing animals for food or editing for human therapeutics, we might be looking at decade or multi-decade long timelines, right? So all of those factors determine where the technology will go first.
One of the things that we often talk about in these kind of forums, obviously, is application. But, before there's application, there's basic research. And Jennifer, you obviously straddle that divide, and you say in the film, you got on this when you sometimes wondered whether you were on a dead end of a field. And one of the things we really wanted to do in this film was to show how the process of exploration, searching for knowledge's sake, can lead to advances that you can never predict. How do you, when people talk to you about all the different applications, and we already have a question about where we're going to draw the line, and I hope other people add questions to the Q&A section. How do you try to weave in this story of the basic research? Because, we live in a time when basic research, the funding, the support is under attack and its relevance, but yet this really is a story that begins with some very unusual pathway and serendipity of discovery, is it not?
That's one of the things I love about the story of CRISPR. I think it's just a great example of how fundamental research conducted for curiosity-driven science can go in unexpected directions, and that's absolutely the case for this technology. While you were asking the question, I was flashing back in my mind to when I was a graduate student. I think I was probably a second year, graduate student, and I had arrived in grad school, just loved science and wanted to do cool stuff. I didn't know exactly what. And I was working in a lab that was studying evolution, molecular evolution, and doing experiments that were ultimately designed to try to answer something about the question, what's the origin of life? I mean, it's sort of a huge question. And I had a little bit of a crisis, when I was sort of in the middle of my graduate career, because I suddenly kind of started thinking, why am I doing this? I mean, it's fun stuff, and I'm having fun, but is that okay? Like, is it okay to have fun in the lab, or should I actually be trying to cure cancer or, do something, quote, useful? And so I went to my advisor and I said, why do you do what you do? I mean, you're obviously doing brilliant work, and you're a tenured professor at Harvard and very, very accomplished scientist. And he looked at me and he said, Jennifer, it's fine to do work that is just designed to answer fundamental questions about our planet, about life. I think that is a great reason to do research. And I've never forgotten that, because it kind of resonated with me, and I sort of felt like I finally got it. But nonetheless, like many scientists, I think as I went through my career, I was always trying to kind of look for ways to make some kind of connection to real world problems. And ironically, CRISPR was kind of my one little area of research that I carved out thinking, well, this will just be pure fun. There's nothing useful here. It's just, I'm going to have fun with this and carve off a little bit of money to work on it. And that was the area that, in the end, probably in my career will be the most useful thing that I've done. I think it's just a great example of the serendipity of science.
In fact, how do you, you yourself as a researcher, how do you walk that line? I mean, obviously the applications for your research are very evident in terms of real world, and the way that Alta talks about the needs are so evident. But yet, there is a need also to just get some fundamental understandings. And, how does one balance the drive for sort of real world product with just the search for knowledge, and how does one lead to the other? And then also the follow on the back of the loop, how does sort of application, how can that also then drive basic research, especially in regards to CRISPR, do you think?
Yeah, there's a lot to unpack there. In my case, the primary interest is fundamental biology, in fact, evolution. So I'm most interested in plants in general and how they make flowers, where they make flowers, when they make flowers. And that, of course, is important in agriculture because flowers drive fruit and seed production. And this question of evolution is something that's very fundamental. There's really no immediate practical value, but we know in agriculture that evolution and evolutionary change is what drives the crops that we eat, right? So the changes that were important for corn to be corn or for tomato to be a large fruit are because of evolutionary changes that naturally occurred and then were selected by humans thousands of years ago all the way up to the present day. So it's an inexorable link between the two, and the desire to study those genes, what we call pathways and networks that control these traits that are important in evolution, either for adaptation in the wild or in the sense of first stages of agricultural production of these crops, from their wild ancestors. These are just connected, inevitably. And in terms of, when things lead to application, they inevitably feed back to the fundamental because you're always getting surprises. So, there are a number of surprises that I won't go through here, but we do some things in terms of application. I work primarily on tomato, and we'll make these genome edits of a certain gene in one variety of tomato and then another variety, and you think that the outcomes will be the same, and they're different. So why are they different? And there's differences because there are other natural mutations that exist between the two varieties that you didn't see and didn't know about, but the genome edit that we created revealed those. And so that leads you down a whole different path of discovery.
And when you talk about sort of this surprise and the serendipity, and really a sense that we really try to put in our film too, a sense of humility and modesty among science about how much we really do know, and sort of how hard it is to predict where we're going to go, and especially around questions of evolution, we really butt up to a central question that we try to address in this film. And Alta, I think this is the topic of our first question from the audience, and ask others, please fill out the Q&A form and add more. But personally, I like where you said in the movie, where do you think society should draw and will draw the line in the future? Do you think people will be able to customize or engineer the genetic trace mutations in their own children? And ultimately, no matter how we're talking about this technology, no matter what the audience is, some version of this question comes up. And you've heard it from many forums around the world. And so how do you respond to that, keeping in mind what we've just heard from Jennifer and Zach about sort of the role of basic research, the unknowns of how that of applications around a technology as powerful as this?
Well, for me, I think it's important to think not only about the technology, but what we know about human behavior. And so since about the 1970s, we've seen a series of reproductive technologies that have given people increasing amounts of control over birth outcomes. We had amniocentesis in the 1970s, and then we had in vitro fertilization and artificial insemination in the 1980s, where you could pick your egg donor or pick your sperm donor. By the 1990s, you could diagnose your embryos and only use those that were free of some particular trait or mutation that concerned you. And at every stage, if you look at the literature, you will see that there is a consistent concern that this is going to become widespread, that everyone will start doing this, and that that's going to fundamentally alter the evolutionary direction of humankind. A couple of things. First, no, it hasn't been something that everybody does, and not only because it's unaffordable, and not even only because there are always risks, although here there are very substantial unknowns about the risk factors. that still need to be explored, but simply because in most cases, people don't want to go to that kind of effort for a return that is just not that important. And second, they are more concerned about having children that resemble their partner than they are about having children that are somehow optimized. And we know this because people have been offered free, quote unquote, superior sperm. Superior because of purported intelligence, as if that's inherited. Superior because of attractiveness by some measure. And what we see is people don't take advantage of it in large numbers. Where you do see uptake is where you have devastating illnesses and where people do still want to have a genetic connection to their children, but don't want to have the children suffer with those kinds of illnesses. The second thing is that if you're talking about evolution, you've got to remember just the numbers game. In order to alter the direction of human evolution, the number of people who have to do this and do it in a particular way is enormous. I mean, unimaginably large to make have that kind of effect. And it also tends to presuppose that evolution is supposed to go in one direction, or even that we're supposed to be getting better and better. And neither of those things are true. Evolution's a lot more random. So I'm able to look at the technologies without nearly the level of anxiety that some other people have about the way they'll expand into uses that we find regrettable. There'll be pockets of it. There'll be some people who use it for things that I personally find offensive, but I don't see it changing the fabric of society in a way that is really devastating to our society.
We have a question, Jennifer, for you here, and it touches across something that I think was one of the inspirations and excitement about us as filmmakers tackling the subject is when we talked to the scientists, you early on and to some others, it was very clear that there was not a divide where we're going to have we could do a film where the scientists were on one side in a monolithic way of viewing things and the ethicists were on the other, and we'd pit everybody against each other. And rather that the scientists themselves, as part of the human population, not a special subspecies, were grappling themselves, and you in particular, with the questions of the ethics around this. And so here's a question: what do you think of the role of ethics plays in advancement of evolutionary biology, specifically related to CRISPR, and more specifically regarding the future studies which could involve CRISPR to cure human genetic disorders and other problems arising due to the applications of CRISPR and the ethical implications on a wider society? How do you view the ethics? But I also would like to get a sense of how do you view your responsibility in this community and in this discussion, in discussing the ethics and confronting that in your own research and in the policy positions that you choose to take or not take?
I think it's critical that scientists are engaged in these discussions, and I think it's absolutely critical also to build a global community of discussions around these topics. I think, and CRISPR is one of multiple technologies that are coming along that are going to impact our world profoundly in the coming decades. So I think that building a culture where scientists and technologists who are developing these things are engaged deeply in those discussions is very important. I was educated, at a time when, at least in my experience, students were mostly, kept in silos. We had, we were doing our experimental work, and there was some lip service given to ethics and science, but my course in that topic was primarily about what is plagiarism and things like that, right? It was nothing about, is your work going to impact other people in a profound way that you need to be aware of and thinking about and engaged in. And so for me, when CRISPR came along and our early work in this field, it became clear that this was going to be a really transformational technology. I had to sort of forge a path that I, didn't have any experience on. And fortunately, we have people like Alta, Charo, who have been leaders in this area of really thinking about the ethics of technologies, and especially in the area of reproductive technologies. But, and Zach, in your field also, right? It's been a long history of misinformation, frankly, I think, around the way that plant breeding is done and what the outcomes of those experiments or efforts in breeders' laboratories are and what the impacts are. So, for me, I guess I just, I've been really trying to, first of all, learn about some of the ethical challenges that scientists and all of us really have faced in the past around technologies and their applications. And then going forward, looking for ways to build transparency into our system, to look to build as much as possible kind of global scientific consensus around responsible use of CRISPR technology. There's another question that is really related to this, in the sense that will it be possible to have global regulations around the use of CRISPR? I feel there won't be. I don't think so. I think maybe the best we can hope for is to have a community of scientists and prominent people in the field who speak up and are in alignment about this. But ultimately, I think that we won't be able to really fully control these technologies going forward. Does that mean we should just ignore it and carry on in our labs and say, well, that's somebody else's problem? Absolutely not.
One question that we get a lot, and Alta, I want to address to you here, is sort of, this is science is a global enterprise. We're seeing with the pandemic how different global responses are within different countries and societies, and how different places approach science differently and draw limits differently. We're also at a period of incredible social unrest and rising, questions about totalitarianism and fascism, and all that carries long histories around, obviously, genetic manipulation and judging people and classifying people around genetics. Sort of, it was a question here about how do you think this can and will be regulated, in terms of a global community, and in light of what Jennifer says, about how hard it's going to be controlled, and how do you see that playing out? I mean, I think this is a question you get a lot, we get a lot.
So Jennifer is absolutely right when she says that it's difficult or even almost impossible to imagine a truly global governance system. That said, it is possible to try to build frameworks that let each individual country, using its own cultural values, adopt a system that's more responsible than simply having no regulation or no law at all. And the World Health Organization now has a committee that's trying to do exactly that. Basically, give governments guidance. And I'll talk now only about human therapeutics. If you're going to approve human therapeutics for somatic editing, do you have an agency capable of doing the following things? And you talk about the kind of evaluation of preclinical evidence, the management of clinical trials, the oversight and surveillance needed to look for side effects. And if you don't have that, then are you able to partner with an entity that does? And are you prepared to think about the following questions? How are you going to distinguish between things that are therapeutically important enough to do, even we're talking just somatic, versus those things that people might call cosmetic or enhancing and are less compelling? How are you going to manage cross-border differences if a patient from another country comes to your country because your country is more liberal, or vice versa? One of your people goes to do something that would be illegal at home, then they go abroad to do it. And if your scientists want to travel across borders to do research that is or is not legal, how to anticipate these kinds of things? But we can't expect every country to have exactly the same rules and the same values. And that goes doubly when it comes to heritable forms of editing, in which we are going to have many countries that simply say it is criminal act, and already do. And a lot of countries in Europe already say that. So it's going to take time to see how the regulatory system develops. Layered on that is exactly what Jennifer said, the real need for the scientific community to be organized well enough amongst themselves to be able to call out somebody's work if it seems problematic and bring everybody's attention to it early on, and to have a venue where they can debate this and educate one another about their concerns. And there is an international commission that's going to be reporting out in September that's looking at only heritable forms of editing, but is also looking very specifically about how the scientific community can self-organize. So we're beginning to make progress, but it's very incremental. We haven't even touched, Zach, I'm sorry, on the complexities of genome editing versus transgenics when it comes to crop regulation, but I'll let you tackle that if you like.
I want to jump in there because I want to turn this discussion a little on its head from what we normally hear. When we talk about, and we have a question, for example, about, it says, isn't the risk that we can and therefore will identify a deleterious gene that can be fixed with CRISPR that turns out in the future to be advantageous? And the specific question here is about sickle cell, which we addressed, which obviously confers some protection to malaria, and we talked about that in this film. It's a wonderful example of David Sanchez and his very inspiring story and sort of his own perspective on this question. But I think that we've already established that this stuff is coming much more quickly in agriculture. We have examples of a GMO debate around agriculture, and that in some ways, we've also heard how hard it is to really change human populations on scale, where you're really affecting evolutionary trends rather than just individual, sort of variation on a more micro level. But it seems to me that when we talk about deleterious genes, when we talk about our own humility, that really in agriculture, I really want to sort of probe this question in its agricultural implications. We have another question about, having cows make better milk or something like that. What kind of questions and humility do you think we need to do around genome editing in the field where you're studying it, Zach, where it's in some way much more, maybe it could get out more quickly, or it might not be noticed as much, or has a much more quicker turnaround time in terms of how something can spread and at the level of human genetics. So how do you sort of look at these questions about sort of risk and benefit and our own level of knowledge?
Yeah, that's a good one. One thing I'll start with is saying that in agriculture, mutations that are considered deleterious in nature are often advantageous in agriculture. So there are a number of cases where there are what we call domestication syndromes. And really what that is saying is that there are substantial modifications to traits that would not be adapted to the wild. But for agriculture, they're wonderful. High yield, compact plant, rapid flowering, various other characteristics. And these are mutations that are desirable, and they are selected for, they have been selected for, and they continue to be selected for. And we've learned over the last 3-4 decades, many of these genes that are responsible for these modifications that would be deleterious in the wild. What I think also is relevant here is the fact that genes and the traits that they impact are not in this vacuum, in the sense that they're often affecting multiple traits. They act what we call in genetics pleiotropically. So they'll affect leaf development, they'll affect the root architecture, they'll affect their interaction with the day length of wherever they're being grown. And oftentimes we have this assumption, and this is where I think your question is relevant for the unexpected. We have this assumption that this particular change that we're going to make by genome editing to 1, 2, 10 genes will result in these predictable trait modifications. But there's so much we don't know about genes and their roles in various aspects of development that we have to be, again, humble and expect that we're going to find things that are not going to be that advantageous. But here is where I say this is not necessarily an issue that should affect regulation, nor should affect humans' concern about consuming genome-edited crops, because this is where breeders and the savviness of breeders and the artistic approach that breeders take to improving crops comes in. They will select against things that are undesirable. They will immediately say, this genome edit you made, you think it looks good for this trait, but now I have a problem with that trait. It's gone. We don't need it anymore. And so I think the breeding side of things will never go away. The very traditional breeding approach infused with the genomics technology that we have and the genome editing technology become a menagerie of tools that basically will self-reinforce and select the best modifications, negative consequences.
As a follow-up, do you find that what you see in the field brings any sense of pause or perspective about using this technology at this point in humans? I mean, or are there any lessons from what you're learning there that can give us a sense of what some of the struggles might be, or some of the hurdles might be in terms of, I mean, obviously it matters a lot less if 1 in 10 tomato plants dies because of an off-target effect or something like that, as opposed to a human at that level. But 1 in a million would be considered, unacceptable. So I'm just curious what you're seeing, what do you see in the field, literally, that might give relevance to this larger discussion in use in humans?
Yeah, so here's the joke. So we walk down the rows and we're looking at our genome edited plants, and we see something we don't like. We look to the left, we look to the right, and we go down and we rip it out and we throw it to the side. So it's pretty easy to get rid of these things, and we don't feel ethically bad about this. This is part of the wonderful thing about being able to work on plants is they're ready and amenable to these types of modifications. I think it's more a question of, still we're in the infancy of just understanding the traits that we want to modify. And what I mean by that is that genome editing is often being pitched as a way to have plants be adapted for climate change. There's so much we still don't know about how plants themselves are adapted and interacting with their environment, and the many genes that are involved in this, that we don't have a clear handle on the targets that we want to go after. So what we're doing are these baby steps, and they look impressive because visually they're beautiful and they're impressive, and they really can tell a story of what's possible. But on the ground, it's a lot more complicated than it seems. And I would say for Alta and for the question of humans and human evolution, changing traits that are polygenic, in other words, involve multiple genes, we're still way at the infancy of being able to understand which genes we would be able to modify. And so I would agree with Alta that when it comes to disease and correcting deleterious mutations, we're going to make a lot more headway there. But the likelihood of us being in a position in my lifetime to be able to edit genes that would modify these quantitative traits that have multiple aspects of control of various different growth parameters seems very, very challenging. We'll get there, but it's going to take a long time.
Elliot, can I?
This was yeah, please do.
And just add to that, because Zach, when you were talking about looking at plants that had been edited and finding that an edit in one tomato background gives very different results than in another tomato background. They're both tomatoes, but genetically they're just different, and I wonder if you think the same is true in humans. You could imagine that you have a gene that has a known function of some kind in humans, and yet editing it in one person won't necessarily give the same outcome as editing it in somebody else. And I think we already know that, just from observation, that that's true in humans as well. So I think that's also a very powerful similarity between what we're doing in agriculture and what could be true in humans.
Absolutely.
I mean, I think one of the things that we tried to, it was a struggle or sort of a challenge in this film was to we didn't want to overhype things. On the other hand, we didn't want to give a sense of just where we are at the moment. And Zach, you bring up this question of timescale, and you're right. I mean, and especially with polygenic traits and how far off that seems. But we also have many lessons in this film. There's David Baltimore, who, at the Asilomar conference, when they're talking about recombinant DNA, none of them ever thought that they would be in their lifetime see a tool developed such as CRISPR. We've seen with the internet in ways that was a real guiding principle for us around this film, is that we've gotten to the point where the internet is so far entrenched in our lives that we didn't have discussions around it before we sort of thought about potentially all of its implications. So I'm curious how each of you looks at this question of timescale. On the one hand, these are very, very difficult questions to answer. There's so much we don't know. On the other hand, science in some ways goes much faster or much slower. And I think we're seeing that in this COVID crisis. The public is seeing real time how science works in terms of steps forwards and backs and things we think we know, and then we realize we don't. But yet, CRISPR itself is a story of unknowable speed and precision and something where nothing could have been predicted, and then it was. So how do you consider that time scale, Jennifer?
Seems to keep accelerating, at least in my experience. I think I couldn't have imagined back when I was a young student in just learning about biology and chemistry that I couldn't have imagined the polymerase chain reaction at that time. And now that's a technology that, for those of you that are familiar with it, it sounds kind of old-fashioned, and it's woven into essentially everything that we do. It's actually the fundamental technology one of them that's being used right now for coronavirus detection, for example. And, I think same with CRISPR, none of us could have imagined that nature had evolved a system like this, and that it would be discovered and harnessed for genome editing, and yet here we are. And so when I try to imagine where this is all going and what's going to happen 10 or 20 years from now, it's difficult to really predict, because I think things seem to move at an increasing speed in terms of the advancements of technology. And I think I'll just add that I think one thing that is definitely happening right now, and I don't really have a handle yet on where it's headed, but I think it could be really, again, transformational, is the convergence of different kinds of technologies, and not just biological technologies, but, let's say computing technologies. And there's lots of excitement about the interface between biology and computing, and yet I haven't met anybody yet that really has a clear vision for where that's headed. We just all have a sense that there are enormous opportunities.
Alta, I want to follow on another convergence here that's raised in one of the questions, and that's the convergence between the science and business. I mean, obviously, this is a technology that has gone, from the lab to the boardroom, so to speak, at a speed that's been tremendous. I think around genome editing in particular, or especially in agriculture, there's a lot of questions about why and how the public has had this complicated relationship. Some of it may be tied around the role of big business or business practices. A lot of people push back on that and say it's overly simplistic. How does the business environment affect where we are? And as soon as you talk about business, you're obviously also talking about government. And so, this is clearly a technology that is already out there in the commercial space, maybe not applicable yet in many forms, but there's clearly a lot of investment in this. What role does big business, or for that matter, startup, small business, play with CRISPR?
Well, I think to answer that, I may have to go a little bit off on a tangent, if I may, Elliot.
Anytime.
Because I don't think it's just about business. I think it's about the relationship between business and the scientific community. And I want to be very careful on how I say this, but in the 1980s, there was a real sea change in which the Bayh-Dole Act allowed scientists in their universities to retain the intellectual property rights to discoveries, even when they had been financed with public money. And this had tremendous payoffs in the form of many of those startups that you're talking about, because unlike the government, the private sector had much more flexibility, was much more nimble, much more motivated to take these lab discoveries and turn them into something that was worth something to industry or to consumers. So we had this explosion of interest in developing products that might otherwise never have been developed. The discoveries would have been of interest scientifically, but they wouldn't have gone toward a product or a process, an industrial process. But at the same time, because of that, scientists that used to be viewed in the kind of, Martin Arrowsmith from the Sinclair Lewis novel as the kind of white hat, self-sacrificing martyrs to science, pure, you could trust them, suddenly were now tainted by the allegation of a conflict of interest, because now they look to make money out of this. And so it allowed for suspicion to grow around the statements that the scientific community made about the value of certain technologies or the safety of them or the integrity of the studies that purported to show that they were safe, which is what happened in the area of genetically engineered crops And the many studies showing that there's no known problem being dismissed out of hand because there's an assertion that they're tainted by conflict of interest. And it's only been exacerbated in recent years by this growing resistance to expertise and to government agencies as a source of independent expertise. So now where do people go for an authoritative voice? We have an explosion on the internet of things that sound authoritative. They have great titles to their pseudo journals or their websites. And it's very easy for people to get terrible information and not even realize that they're getting it from a completely flim-flam source. So I see a challenge here for us. And I'm not saying we should get business out of the science because of the benefits it's brought us, but we do need to figure out how to regain public trust. And that would be an important first step.
I mean, hopefully that's that was one of the inspirations for this film and our larger effort at the Wonder Collaborative is really try to rethink, because not only, is it flim-flam sources, but there's so much complexity around this and having worked in journalism and seeing even how it's covered in journalism. And I'd say the problems often don't stem around necessarily getting the quote unquote facts right. I think that science is by its nature, there's a culture to science, and that culture is poorly understood. Most journalists, even the ones who cover it, haven't worked in labs. My father's a scientist, so I grew up around it, but I think that we're one of the issues that we're seeing with the COVID-19 coverage, even in the press, is a real misunderstanding about how science works, the incentives around science, how that people can get things wrong in good faith, that first answers, that we have incomplete knowledge. And one of the things we try to capture in this film is to try to put a marker down with the public. And a lot of the distributors around the public who think, the public's not interested, or you can't make it complex, you can't make it nuanced, you have to dumb it down. This is one of the things I want to, it really wasn't so inspiring from our early conversations with Jennifer, was that, you refuse to do that. I think you've been lauded rightfully for the power of communication and the importance you place on communication. But I would say it really must start with a respect for the public that you feel, and I think others, and hopefully, and the people that we profile in the film, I think all came together and believed, look, we should lay it out there. And we're very excited. I'll do a plug for the film. It's going to be on NOVA on September 9th at 8 p.m., a special 2-hour NOVA. It's going to be on Netflix on October 1st. And so, we've played film festivals around the world. So I really do think that there's a good news story that if you treat the public with respect and you trust the public, the public can actually deal with very complex science. So maybe, Jennifer, there is a question about how did you get to be such a wonderful speaker about science? Maybe to get at that, how do you approach communicating this topic to the public? What is when you think about messaging, do you have any certain guidelines that have driven you in terms of the way you talk about it?
Well, again, it starts for me with a little reminiscence from graduate school. So my parents came to visit me in Boston, and I was so excited to show them the lab. And I was just so excited about my research and what I was doing and experiments. It was all new for me, and I just thought my parents were just, think this was great. So I brought them to the laboratory, and the first thing that happened was that my parents said, "It really smells bad in here." And I couldn't but I realized, no, it's because we grow E. coli bacteria all the time. And yeah, they don't smell very good, but I didn't notice it anymore. It didn't faze me at all. It was my work environment every day. And then, so my parents wanted to know, so what do you do all day? And I said, well, I'm studying this really important problem. I'm trying to understand the molecules that might have led to the origin of life. It's so interesting. And they said, well, how do you do experiments? How do you see the molecules? And I said, well, we don't see the molecules. Molecules are in these little droplets of liquid, clear liquid, and we test for their presence, using different kinds of chemical types of tests. And so my mom looked at me and said, you mean you spend all day moving droplets of clear liquid from one tube to another? And I realized, yeah. So, if there's a lesson there, it really brought home to me that, if you want, I just thought I was over the moon about what I was doing, but I was so far down in the weeds, that I had to really pull myself back and remember what was it like the very first time I went into a lab, and saw what people were doing, and how did they explain to me, why does this matter, and how do they actually understand their data. And so at some level, I've been, kind of working on that problem ever since. I mean, it's a big challenge for scientists, because the farther you go in your field, and I think all of us here are familiar with this, the more we're in our own language. We have our own acronyms and the way that we talk to each other at meetings, and we have to really pull ourselves back from that and try to remember, what was it like when you were learning something for the first time. And I guess the other thing that's helped me a lot is that I've had the great pleasure to be involved with a number of organizations like the Packard Foundation, the Pew Charitable Trust. These are organizations that fund scientists in a variety of different disciplines. And so when you go to those meetings, yes, it's all science, but everybody has to, at some level, communicate their work and why it matters to people who are not specialists. And that has helped a lot, because, you can see how other people do it. Some people are better at it than others. And I've tried to, learn from people who are really, really good at science communication. And there are people out there that are, really very, very good at it. And I think the more that we can encourage our own trainees and students as much as possible, what I tell my own students is you should have a one-sentence answer to the question, what are you working on, that kind of communicates what it is and why we should care.
Well, Zach, I will try not to use, Jennifer used to be down in the weeds. I try not to use it as a segue to your work, but in some ways your lab, part of it looks like what your backdrop is, which is a very different environment. But how do you, in some ways, people have a much more familiarity. You're not necessarily moving clear liquids around, although at the some level, you're also doing the very same thing. How do you view communicating science? And I think it's important what Jennifer mentions, that, that for scientists, once they get out of their own specialized field, often communicating to their peers doesn't have to be that different in some ways than the general public. But how do you talk about your research? What approach do you take for that?
I really look at it from the perspective of what still gets me excited. So I'm excited to walk down the field, look at these plants, see these trait variations, and then think about the chromosomes and the genes and everything that's happening in terms of how Mendel and what we learned from Mendel and his P's is now happening with genome editing and what we're doing. So my communication, which I actually do quite a bit with elementary school students and middle school students, is to just show them how cool science is. I mean, really, I know it sounds trite, but it really is very cool. And the more I can bring them to that level of understanding of how cool it is, I think it's much easier for me to get them excited about some of the nuances that maybe they would be bored by in a classroom. So for me, it's always about trying to talk to them in a way that gets them excited about what's really innovative and cool about science and biology overall. And that seems to work pretty well. At least it works with my kids, most of them anyway.
Right. And we just have a couple more minutes, Alta, but you're a wonderful communicator yourself. How do you, and your communication of the film was vital for it, really giving a sense of this technology and where the limits are. How do you view your own role in communicating?
I spend a lot of time doing exactly what Jennifer was talking about. Know your audience. Talk to people about what they want to talk about, and then you can throw in the stuff that you find really interesting that they may not have thought about yet, and they may come along with you if there's real passion and real enthusiasm in the way you address it. But you have to start by talking to people about what they're interested in, as opposed to just scolding them and telling them that they ought to be interested in what you think is cool. But I couldn't agree more with you, Zach. It's because of the cool factor that we can get people to want to explore this out of curiosity and out of wonder.
Well, thank you. And that's a wonderful plug for our Wonder Collaborative. And that's the reason why we called it that. It's the production behind this film. And I want to thank all the panelists for joining us and Folsom Harbor Laboratory for hosting this. once again, I hope you enjoyed this discussion and human nature. Please, as I mentioned, it's going to be viewed on NOVA on September 9th at 8 p.m. And also then it'll hit Netflix on October 1st. So please, watch it. The more people that watch it and believe in this kind of science storytelling, the more hopefully others will invest in it and let us do more projects like this.